folder in the YouTube, so you can check the link to go back and look at the video and actually go to the list. You won't have to sort through which one is my class, and look for your fourth period video assignment in the YouTube. So you can check the link. Okay.
If you have a child 12 or older and you'd like for them to get into the best university possible, I have something very important.
Okay. All right, just waiting for all these folks to come in. In the meantime, look at Canvas. Canvas, again, we are in the CR module for mitosis. So we're almost all here. All right, good morning, good morning. Again, we're in the CR module for mitosis on Canvas. So we're gonna hop back and forth between Canvas, and I have a video for you to watch. Also have a piece of paper out. I need you to draw something today. So we're starting cell reproduction, new unit, and there's a lot of notes that you need to take. This one is important for you to take notes. Very important for you to take notes. Okay. I'm going to go ahead and pull up GoGuardian, then the attendance, and then we'll get started. But in the meantime, like I said, be in Canvas, looking at these. If you haven't looked at the vocabulary on the first page, make sure you look at that. It's a lot of vocabulary. Then go to the second page for mitosis. And I will do attendance, and then we'll get started. Still have some 2 or 3 people missing. Hopefully they'll get in. And then I'll give some housekeeping conversation once everybody's in, so I don't have to repeat myself. Okay, cool. Everybody's here today. That's wonderful. All right, so that's good. So then when I say this, I said this to everybody one time, and hopefully we don't have to say it again. All right, most of you are very punctual. I got Natalie, you're always my first person in class, so kudos to you. But everybody needs to work on being a little more punctual to class so that we can get started. We don't have to waste a lot of time. You do have an end of year exam for this class. So you need to make sure that you're taking notes, and you need to make sure that you're using this time to the best that you possibly can. Secondly, we're asking to remind you all that you all need to be on camera all the time, and you need to be focused on this class. And one of the things with GoGuardian, they can see how many tabs you have open, and whether you're focusing on this class or somebody else's class, or you're playing games in the background. You need to, especially as we go forward, these units are going to get a little more difficult. So you need to make sure that you are focused on this class. And I say that I'm saying that in general because you all are usually very great students. So that's just my everybody's speech. Okay. But it's been said. All right, attendance is done. Everybody's here. Wonderful, wonderful. And again, we're going to try to be punctual next. And so let me share my Canvas screen. Again, if you just came in, we're in Canvas, we're in the CR module for mitosis. Up to now, we have talked about its organelles and the fact that the cell is the basic unit of structure and function. Well, now we're going to talk about how cells reproduce. Okay, so the whole conversation is going to be about what's going on in a cell, but more particularly, it's going to be about what's going on in the nucleus. So after we look at the general structure of the cell, so I have a warm-up activity for you to do, and I need everybody's participation. I'm going to start with this Nearpod. I had hoped that it was going to do some more things, but it went in a different direction. So I just want you to do. Let's, all right, I'm going to send you the link. Okay, why did it not copy? Copied. There we go. All right, so log into that Nearpod. Keep your canvas open, but log into that Nearpod. And we want to look at the essential stages of the cell cycle. That's our objective today. All right, so we're going to start here. And I'm going to give you a few minutes to just label those parts of the cell. So I need all, and that's another thing, part of my new semester speech, need everybody to participate. So when I check the student view to see who's participating, I want to see everybody participating. I don't want to see no answers. Everybody needs to be fully engaged. All right, so notice we don't have all of the parts of the cell labeled because right now we're only interested in a few of the parts. So for cell cycle and for cell reproduction, we're not going to talk about what all the other parts are doing. We need to know what those things are doing. So take a minute to see if you can label those parts. And I will check with you actually, I'm going to stop sharing for a minute and make a correction to the attendance. And mm, the other interesting thing about Go Guardian is it shows resourcefulness. Some of y'all are just looking up the answers. And that's okay. That's okay. That's okay. It's been a minute since we looked at the organelle, so that's okay. And I'm gonna come back to sharing. When am I sharing? Okay. And then we'll talk about what your answers should be. All right. So let's see if everybody's got it down. Let me see if I can pick on somebody who's got it. I look like you're still working on it. It's a toss-up. All right, I'm going to use this one. Okay, Malik, you're almost right. Malik, you have one right that Xylid has. All right, Xyla called this one down here to the bottom lysosomes. And Malik, you had it right. These are the ribosomes. So everybody fix your picture. This one is most correct. So the larger structure is the nucleus. The smaller structure inside the nucleus is the nucleolus. The large red structure on the outside is the mitochondria. And we include the mitochondria in this discussion because all activities require energy. And you have to remember that our mitochondria is our energy factory. So that is going to be important just because that's what it does. Most of our conversation for this unit will just be within the nucleus, what's happening in the nucleus, what's the nucleolus doing. And because we make what we're doing in the, there you go, ribosomes are important as well. Okay. All right, so everybody should, so when I switch back here, I see everybody has fixed their picture. Um, what did I do? Oh, I need to stop, go back over here. All right, there we go. Okay, cool. All right, so that was just kind of a review so that you remember what everything is in the cell. Now, again, we're going to, whoops, that was a question. You, okay. Okay, let me go back over here. Um, I'm gonna skip this question because we are gonna spend a lot of time talking about, we're gonna watch a video and we're gonna talk about what happens as cells grow. So I'm gonna skip this one. You don't have to answer that. But we are going to look at the cell cycle. And I think this is where I stopped using this because they skipped to a video that did not work for us. So you can log out of Nearpod because I really just wanted it for that opening part anyway. So let me stop sharing for a minute. And actually, let me go here. Okay, go to Canvas, let me share Canvas. And there's a video at the beginning, and then she's got a little bit too much energy for me. So we're going to watch the Amoeba Sisters video instead. But what I do want you to know is the definition of mitosis. So if you have, let's go back to the first page. If you haven't written down a definition, let's go start by taking, and it's not on there. It's got meiosis. It should have mitosis. Okay. That's not good. All right, but so this is why we need to write this down because mitosis is cell division. Okay. Simple cell division of body cells. We're going to watch this video here. And then this is where I need. Okay, I'm going to pause it in a couple of places, but like I said, this is where I want you to have that piece of paper out. You're going to draw 2 circles. So when I get to that part, I will pause it and then explain what it is you need to do. Okay. All right. Again, mitosis should be the heading. All right. All right, this is a review also, and she said it weird, but it's deoxyribonucleic. We're going to spend a lot of time talking about DNA. Okay, so you need to remember that it is a macromolecule. It is a type of nucleic acid and reproduction. All right, that's so write down that vocabulary word chromatin, because we're going to have a couple of words that all sound the same. We're going to have chromatin, chromosomes. Um, There's another one, but we'll get to it. And you need to know the difference. So when we say what she said, long strands of chromatin, that's the part where the DNA looks like spaghetti. So when we look in the nucleus and we look under the microscope, it looks like spaghetti and it's dense and taking up most of the nucleus. What she's going to talk about eventually is that DNA is going to start to shrink, and it's going to, when we start talking about chromosomes, we're talking about a condensed version of the chromatin. Okay, so you do need to know the difference between chromatin and chromosomes. So chromatin are the long strands of loose DNA. We'll call it that, long strands of loose DNA. Chromatids. All right, that was the other word that I was looking for. Chromatin, chromosomes, and chromatids. That's the third one. Okay. All right, so if you didn't jot down, chromosomes are the condensed, like I said, version of the chromatin. Now, what she's saying here, in the nucleus, they're going to make a carbon copy of themselves. So once they do that, we call the copies down here at the bottom sister chromatids. Okay. And chromatids are carbon copies of the chromosomes.
Not all cells divide, though. A number of cells in your body, such as nerve, red blood, and muscle cells, do not divide once they are mature. The cell cycle is a timeline of events.
All right, so this is the one I want you to watch a second video, but it's going to explain more of the mitosis to you. But we're going to start with this drawing because when they do the second, so you want to draw two circles. You want to put the title cell cycle in the middle. And you might want to add an arrow that starts with cytokinesis and goes in a clockwise direction. I can't draw on this because it's a video, but oops. Okay, your arrow should start with cytokinesis and go around the outside so that it goes in a clockwise direction so that you know which way this activity is going. So I'll give you a minute to draw that. Make sure you put all the labels in because you're going to have to know what all these parts are. And again, there's a lot of vocabulary with this, so try to stay on top of the vocabulary. Starting with this word cytokinesis. Okay, you can hit your reaction button to say that you're done drawing. Just give me a thumbs up. Okay, I got 3. And if you're finished drawing, give me a thumbs up, a reaction thumbs up. All right, so she's going to explain this and they're going to talk about it again in the next video. But our focus is knowing the phases of cell division. And notice the big green section. Think of this as like a time clock. That big green section indicates how much time a cell spends in that particular phase of the cycle. So that's supposed to tell you that most of the time the cell is in interphase. Okay, during interphase, it's getting ready to do all of prepping the cell and prepping the chromosomes for mitotic division. So the mitotic division, you see you have your P, M, A, T. That's the phases in order. Okay, I'm gonna let this go on because, like I said, we're going to see this again.
During interphase, the cell is getting ready to divide. Interphase is divided into 3 sections. G1, S, G1, the protein division, and has DNA copy of itself, which is DNA replication. G2, cells make copies of the organelles.
Okay, I'm gonna pause it right there. Please make note because you've put those initials on your cycle. So now maybe underneath the cycle, you want to say what G1, S, and G2 are doing. Okay, so proteins are produced within the cell during G1 that will be necessary for cell division. So that's why we had to have our ribosomes because the ribosomes are our protein factory. During the S phase of the now all of this is going on during interphase. Okay, just remember all of this is going on during interphase. All right, so the DNA makes a copy of itself. It replicates. And during G2, everything else in the cell is making a copy of itself. Okay, because when we divide this cell, each cell has to have exactly what the first cell had. So we want everything to be exactly the same. So we're making carbon copies of everything in the cell, but it starts with the DNA. All right, we good? Give you another minute. Okay. Okay, write that down. And this should be your definition of mitosis. Mitosis is a type of cell reproduction that produces identical cells. You want to add to this body cells that produce, this occurs in the body cells, because then we're going to do meiosis, and that occurs in another type of cell. So mitosis occurs in all body cells. Okay, and the important part is this means that the cells are producing the same DNA. Each cell looks exactly like the original. We call it a parent cell. The daughter cells look like the parent cell.
This makes producing cells that have
All right, so she was just getting warmed up. So that's a general overview of cell reproduction. Now we're going to look at the short section of it. So all of that was interphase. That was the big green outer circle. Now we're going to look at that little short part of it called mitosis. Okay, so cell division has two events, the interphase part and the mitosis part. Okay, so let's look at these. And we're going to come back and review with this, but I'm going to share this video first. She's going to repeat some things that you just heard, but she's also going to go through PMAT, which is what we need for you to know from mitosis.
Have you ever looked down, no idea where it came from or how you got it? So you put a bandage on it, and a few days later, it's gone. It's all healed, and you don't even think anything of it. This amazing process that causes this to happen. Or let's say you're looking at your nails, and you notice they're a lot longer since last time you cut them. Or let's say you're looking in the mirror, and you notice that you are a lot bigger than you were when you were 5 years old. What do all of these things have in common? One major thing they have in common is mitosis. Mitosis is a type of cell division done by most of your body cells, and it's really important for cells to divide. If they didn't divide, you wouldn't grow. I mean, how do you grow if you can't make more cells, right? So one reason why you're bigger than you were when you were 5 is mitosis. Mitosis also is great for repair of damage. If you have some kind of accident, like we were talking about that cut on your arm or leg, well, you want to make sure it gets repaired well. So you have to make more cells to do that. Mitosis is great for that. Now, it's really important to understand what it is not. Mitosis is not a process that makes sperm or egg cells, because that's something different called meiosis, which sounds like mitosis, unfortunately, but it's a different process. Mitosis is done with body cells. Mitosis makes identical cells. That is the goal, identical cells. So if you're trying to make more skin cells to replace worn out or damaged skin cells, you don't want to start suddenly making stomach cells there. That would be ridiculous. You want to make sure you have identical cells replacing what was lost. So mitosis makes identical cells. It's a really important thing. Now, it's also important to understand that your cells are not divided. If all they did was divide, it would just be rapid, crazy growth. In fact, on the cell cycle, actually most of its daily life, which is actually a phase called interphase, where it's growing and DNA, carrying out its daily cell functions.
Okay, I paused it there and I'm gonna take the closed caption off just for a minute so you can see that. All right. Basically, next to your G1, your S, and your G2, you want to write growth, DNA replication, and cell function, because that's what those represent. So in a nutshell, if you had to summarize what is happening during interphase, it is growth during the G1, DNA replication during S, and cell function during G2. Okay? I'm going to ask you later what that means.
That's where cells spend most of their time.
Everybody should be on camera.
Mitosis is a very short amount of time in respect to the whole cell cycle, but mitosis is a critical process because this is where it is going to divide and make more cells. Before we get into the subdivision, it's really important to understand that more cells have something inside them, an organelle called the nucleus, and the nucleus holds your DNA. DNA is really important because it's your genetic information. And if you're going to make more cells, you need to have the same DNA in those new cells as you did in your original cells. You want it to be identical, no mistakes. Very important. The problem is you've got a lot of DNA. We've got to get that DNA into the new cells using mitosis. So there has to be a better way to organize that DNA. Well, what actually happens is that DNA can be organized into these condensed units called chromosomes. Chromosomes are made of DNA protein. You've probably heard before that humans have 46 chromosomes. That means 46 chromosomes are found in most human body cell nuclei.
All right, I'm going to pause it right there. All right, so that's something that you need to remember for humans. One of the reasons we can identify look at a cell and identify one cell from another cell, because there's everything has a unique number of chromosomes. Humans have 46 chromosomes. Onions might have 12. Elephant may have just a random number 50 because they're bigger. And don't quote me on those because I don't know the exact number, but the point is every organism has a unique number of chromosomes. And you're going to need to remember that number, 46, because when it duplicates, it's going to double. And then the other thing that you're going to have to keep in mind is there are 46 individual chromosomes, but they hang out in pairs. So we tend to say there are 23 pairs of chromosomes. So again, that number 46 is important. And the fact that they run in pairs is also going to be important. All right. And she also mentioned that chromosomes have that protein structure holding them in the middle. So again, chromosomes are condensed chromatin material with held together by a protein.
What are nuclei? Well, it's the plural of nucleus. You don't say nucleuses, you say. In the nuclei, there are 46 chromosomes. Organizing DNA into condensed chromosomes makes it a lot easier to move over when you're making new cells. So if you have 46 chromosomes in a human body cell, you have to duplicate those chromosomes in interphase before mitosis starts. That basically means you're duplicating your DNA, since chromosomes are made of DNA protein. You have to do this before mitosis starts because if you're going to make an identical cell that has 46 chromosomes, just like the original, well, it makes sense you have to duplicate the genetic material before splitting. So if you look at our cell cycle video clip, we talk about interphase. That's a stage where most of the time cells are spending their time. They're actually duplicating their DNA during that time. So ready for the tricky part? Because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes, as the sister chromatids are still attached, and we're counting by centromeres. So 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture, but you went from 46 to 92 chromatids. video explaining that in more depth and how that factors in more mitosis. Okay, so now we can get right into mitosis. I like to post students to remember PMAT. It's a little acronym that helps you remember. The P is for prophase, the M is for metaphase, the A is for anaphase, the T is for telophase. So remember PMAT, the stages in order. The very first step is prophase. Prophase because it's the beginning step, the nucleus is still there. And it's going to go away later on, but this is a stage where it's actually still there.
All right. What's going to happen, and then you're going to have a matching activity in a little while. The important thing is what is happening to the nucleus. So right now you need to remember what is happening during each phase. You're going to be quizzed later on where they jumble everything up, and you're going to have to match what is happening with the title of the phase. It might be the picture, and based on what the picture is showing you, you need to remember what is happening. So with prophase, you need to remember that the nucleus is still there and the chromosomes are kind of still all over the place. Okay? So remember, as you watch this, remember the events that are happening in each phase.
The chromosomes are visible. In fact, we say they're condensing, which means they are thickening and visible. The next stage is metaphase. M for metaphase, but I also like to M for middle because in this stage, the chromosomes line up in the middle of the cell. The nucleus is disassembled, it's no longer there. So we've got the chromosomes in the middle waiting there. Next, the A is for anaphase. In anaphase, I like to think of the A for away. The chromosomes move away. They are moving to opposite sides of the cell, so they are moving towards the poles of the cell. Now, one thing to point out, these chromosomes, they're not moving by themselves. They actually have something called spindles. These spindles are fibers that help move the chromosomes to the ends.
All right, and we'll stop it there. All right, so there's a couple of things that you need to know as far as vocabulary. What is a centriole? The centrioles are structures in the cell that create the spindle fibers. Okay, then the spindle fibers are attached to the chromosomes. And remember how they were two attached by a centromere in the middle? See, all these words kind of sound alike. Centriole, centromere. So you're going to have to make sure that you clear them out in your head. All right, so the spindle fibers are connected to the chromosomes, and they're pulling, they shrink. So as they shrink back to the centriole, they pull those pairs apart. Okay, so that's what you're looking at. Now they're back to being single chromosomes instead of being chromosome pairs. All right, so again, make sure you're checking vocabulary. You need to know what a spindle fiber is. You need to know what a centriole. What do they do? What is their job?
Kind of helps them move along. The last stage of mitosis, think T or telophase. In telophase, the chromosomes are actually at the complete opposite ends, and new nuclei are forming on each side to make these two new cells. The nuclei are starting to surround the chromosomes on both sides. I like to think of the T for 2 because you can really see in the step that the end goal is going to be 2 cells. In the human body, they're each going to have 46 chromosomes. And again, remember, they are identical. Now, cytokinesis is responsible for the final separation into 2 cells by splitting the cytoplasm, which completes after the PMAT stages. So why did all this matter? Without understanding cell division, we wouldn't understand how growth and repair happens, because they both require more cells to be made. Understanding mitosis is also very important for cancer research. Cancer itself is uncontrolled cell growth. So in other words, uncontrolled mitosis. Well, that's it for the Amoeba Sisters, and I'll invite you to stay curious.
Okay. All right, so again, there's a couple of things we need to remember about that. All right, so let's come back to Canvas, and they're going to summarize all of those events right here. All right, so we've done interphase and we prepped the cell to divide. And then we have our phases of mitosis. Now up here they have just PMAT, which is wonderful. Because down here on this chart, and I say that because down here they added an extra piece, which kind of just extends what's happening during prophase. But if you click on each of these, it actually describes what is happening during each phase. So you need to familiarize yourself with visually and be able to say in words what is happening when you see this picture of the cell. So again, prophase, you can think of P for prepping the cell. So the chromosomes are condensed and visible. Like I said before, they look like spaghetti. A lot of times if you scope, and I'm going to try to get you all to look at cells alive again so that you can see what an actual cell looks like and watch it go through these stages of mitosis, where the initial cell, you know that it's going dense center to the nucleus. And eventually, you know that it's prepping for prophase because you start to see chromosomes, but it's still surrounded by a nucleus. This is where the nuclear membrane starts to break down. Okay, so this is the important, I don't know if I can highlight that right there. This is the important part about recognizing that the cell is in prophase. This is where you see visible chromosomes and you see that the nuclear membrane starts to break down. Okay, all right, so that's P. All right, during the M stage, they say M is for the, think of the middle, and they want you to think of the middle because the chromosomes line up in the middle. Okay, the chromosome pairs line up in the middle. Okay, and that's the biggest issue. Two things actually. The chromosomes line up in the middle, and the nuclear membrane is gone. There is no nuclear membrane in the metaphase. It's gone. And the spindle fiber. So three things, I guess we can say, happen here. The spindle fiber up in the middle, and the spindle fibers attached to the chromosomes. Okay. Wake up, Lana. All right, next phase, anaphase. And she said, think of A as a part, but this is where the spindle fibers begin to shrink. And as they shrink, they pull those carbon copies apart. Okay, so they pull them away from each other. But as they shrink, they go to opposite sides of the cell. And the last phase is telophase, where things start to try to get back to normal. So you've got the chromosomes on opposite sides of the cell. Now we need the nuclear membrane to come back. And then everything else, we said that during interphase, the other organelles are duplicating. Well, those things need to go to opposite sides of the cell as well. Okay, but we don't number those like we do. We don't say that we have to have 5 mitochondria. We need 5 mitochondria over here. We don't worry about how many mitochondria. We do need to know how many chromosomes, because the chromosomes are, the DNA is the boss of the cell. It tells it, the rest of the cell what to do. So once we get that exact number copied, everything else will fall in line. So we're really focused on making sure that we have exact copies on each side of the cell. And then that's the end of mitosis. Okay, so they say PMAT. So you notice this box is around the prophase, metaphase, anaphase, telophase. Cytokinesis is the last stage, but that's a separate stage because all it's doing is just dividing the cytoplasm and everything else. Okay, but mitochondria is duplicating the DNA and separating the DNA and creating those 2 sets of DNA. Cytokinesis is just dividing the cell. Okay, so it divides the cytoplasm and all the other cell contents. Okay, all right. So down here again, you need, if you're breaking a note, cell division equals mitosis plus cytokinesis is a good note to write down. This is okay, and this again is going through the stages. Actually, it goes by itself. All right, it's playing, but it has no sound. All right. And again, looking at the during the S phase, we are making copies of the DNA. Okay, there is no sound on this, so you kind of have to read your way through. But do use this as a review. I did put the Amoeba Sisters video in the YouTube folder for this class, but you can easily use this one as a review as well. And again, they describe what happens during each phase. All right, so this one is anaphase. They pull the cells away from each other. Telophase is when the nuclear membrane is coming back. I'm trying to speed this up a little. And then last, cytokinesis. All right. All right, so again, down here, they have given you a visual. And down at the bottom, if we go to, next time I'll let you look at cells alive. If you were actually looking at a real cell under the microscope, that's more what it would look like. They kind of illustrate it up at the top, but it would look more like the pictures at the bottom. But you would still be able to look at each. And if you're looking at a cross section of sometimes they use onion skin, and you can identify different cells doing different things at different times. So all cells don't do the same thing all together. This cell will start to divide, maybe the next cell will start to divide, but so you can identify what phase of mitosis is going through by what the chromosomes are doing. All right, so I want you, the other question, we said this happens in plants and animals. So the first question is, do plants and animals reproduce the same? Yes, they do. They do. Plants have a branching in. With plants, they have a plate that grows in the middle and it grows out, and because they have a cell wall. So they have a plate that grows in the middle, spreads out, and then the two cells divide. But they still do PMAT. They still have interphase and PMAT. Okay. All right. The other thing is they don't have centrioles. They have microtubules, but the microtubules do the same thing that these centrioles do. They are responsible for grabbing those chromosomes and pulling them apart. All right. I want you to take a minute, everybody, and do this section right here, drop and. So you want to grab, and then you can use this chart up here to help you. So you don't have to be on any other page. You don't have to be looking up stuff on Google. Just go scroll up here to see what it should be. And you're going to, this one says each side of the cell has a copy of DNA. And that's so each side. So, and if it's not in the right place, it won't stay. So you're going to grab the phrase and put it in the box where it belongs. So everybody take it if you're doing it. Landon needs to wake up so he can do this because he's here, but he's not here. All right, everybody again needs to be on camera. Some of y'all, I just see the top of your head. Ariana, I haven't seen you in a little while. All right, so again, drag each word into the place where it belongs. Back so well. Join us. We're all doing a drag and drop. Like I said, you can scroll up to the one at the top and look at the box up at the top to see what's going on. I'm gonna have a little matching quiz for you tomorrow, just similar to this, where you will have to match the picture with what's going on. So this is practice. Okay, everybody should have completed that. And you should have given a check. And you notice when you finish, so all of your, I noticed it wouldn't go in, it wouldn't stay in the box if it wasn't right. Okay, when you finish, whoops, I didn't mean to do that. Just check the check and it'll tell you whether you're correct or not. Okay. All right. So again, this is in summary, you need to know that there are two phases to the cell cycle, interphase and mitosis. Interphase is 90% of the time. Think of that drawing that you have as kind of a clock that it spends most of its time during interphase, that it's prepping for mitosis. So the it's growing. The cell is growing. The cell is duplicating its DNA and it's getting ready for um, mitosis. All right, you need to know your, uh, most of the time people don't do prometaphase, but um, just know PMAT. PMAT is prophase, metaphase, anaphase, telophase. That is mitosis. All right, and it's going to be important to remember those because again, we're going to see this again when we do meiosis, but we're going to add to it. All right, then you need to know that cytokinesis is the last stage. Okay, so it's actually kind of 3 stages. I said 2, the interphase, and then interphase, mitosis, and then cytokinesis. That's a separate part. All right. All right, I don't want to start meiosis yet because that's a whole different type of cell division. So what I do want to do is, give me a second, in the last few minutes, any questions about cell cycle? All right, we'll see. Tomorrow you're going to have a little quiz. So I'm going to see if you were paying attention and the 3 of y'all was taking a nap. We will see. We will see. All right, so I'm going to go back and look at this Nearpod, but we're going to skip a certain portion of it. So I'm going to put that link back in the chat. Same one. So we're going to continue from where we left off. All right. And I want you to be here. All right, so everybody should be here. Everybody should be logged in. I need you to answer this question, and I need you to answer that question. Okay, so answer those 2 questions. Let me check and to see if everybody's awake and participating. Because right now I see some folks missing and out. Oops, okay. Oops, I think I sent it to one person instead of everybody. Hold on. There we go. All right, so you have a question. So you skip ahead so that you're at this screen, slide 14, and answer this question. What do you think it is? What might the cell be doing during this phase? That's the question you need to answer. And actually, I should have sent this to your student. Tell you what, let me do that. No, I'll leave it. Take a minute to answer that question because we only have 4 minutes. So 2 minutes on this one, and then I'm going to advance it to the next question, and that'll be all we'll get done today. So I need an answer from everybody. Remember, this records whether you participated or not. Participation counts. And I'm going to be looking for that more this second 9 weeks. Are you participating? Are you turning your assignments in on time? Last 9 weeks, I excused a lot of latenesses, but this 9 weeks I'm not. So when an assignment is due, it's due. And because what the grade book does is if it's late, it takes off automatically. So some people had zeros for things that I graded because it was submitted so late that Canvas said, "Up, that's late, it's a zero." So I had to go in and take the late off in order for the grade to show. I'm not doing that this 9 weeks. So you need to stay on top of things. You need to be on time for class, log in, be on screen, and submit work when you're supposed to submit it. All right, one more minute on this question, and then I'm going to advance it to the next one. So I need to see an answer from everybody on what is interface? What's happening during interface? All right. Hopefully you had an answer of some sort there. I'm going to go on to the next question. Everybody good? Good. Finish your sentence and I'm moving on to the next one. All right. What are the objectives? What was the point of today's lesson? All right, so I'm gonna go back over here. Look and see what you said. Maxwell, you got no answer. Everybody answered. All right. So interphase, the phase of the cycle where, how do I get to see the rest of it? Okay. The cell prepares growing. Okay. Interesting. All right. Again, study your vocabulary, folks. Study your vocabulary. All right. You all have a great day. I'll see you tomorrow and we'll go through a little quiz.